among many other things, in the field of infectious diseases, and trying to understand the temporal and spatiotemporal dynamics of ecological processes. We finally have Dr. John Balbus. He is the Senior Advisor for Public Health to the Director of the National Institute of Environmental Health Sciences, where he directs the NIEHS-WHO Collaborating Center for Environmental Health Sciences. He is trained as a medical doctor and has extensive experience working at this fascinating intersection of health and climate. So just as for you to know which will be the structure of this panel session, we will now begin with brief opening remarks from each panelist, and then we will proceed to questions. So a number of questions have been prepared in advance, but we will also reserve time for questions from the audience. So you have, if you have questions now, just as the session evolves, please type them on the Q&A box on your screen. I also note that any other questions that we don't get to live will be entered to the AGU Connect discussion site for the symposium, where we can continue the exchange of ideas after the session. So the time for each panelist will be strictly limited to 5 minutes. So I expect you to make very compelling remarks and ideas on the topics of discussion. So I pass the floor to Anna as the first speaker.
Okay. Thank you and good morning. Hello, my name is Anna Stewart-Ibarra. I'm the Scientific Director of the Inter-American Institute for Global Change Research, an intergovernmental organization that serves 19 countries of the Americas. Thank you for the opportunity to participate in the panel. So although the direct effects of climate on COVID are still being disentangled, I wanted to share 3 observations. First, the pandemic has revealed and deepened social inequalities and capacity gaps around the world, particularly in the global South, resulting in cascading climate and health impacts that go far beyond COVID. What happens when people are forced into quarantine in dense urban slums without adequate access to water, food, and basic sanitation, a situation that is exacerbated by extreme climate conditions? In 2020, the UN Economic Commission for Latin America and the Caribbean, CEPAL, projects that the number of people in poverty will increase by 45.4 million in Latin America, representing more than 37% of the population. How are we going to address the growing inequality gap and the resulting effects of climate on COVID and related health issues? Second, we need to understand the two-way feedbacks between COVID and climate. What will be the impact of COVID on climate change? For example, under growing economic and political pressures, some governments in the Amazon region are reducing environmental regulations, while illegal mining and deforestation activity is increasing. There are projections that forest fires could be worse this year than last year due to dry climate conditions. Loss of critical forest will accelerate climate change while increasing respiratory health complications for populations across the region. Researchers are investigating this nexus and working with local communities, schools, and decision makers to develop online monitoring and warning platforms. Finally, this pandemic provides us with an opportunity to bring together the climate and health communities to create a more cohesive response to climate-sensitive infectious disease outbreaks today and in the future. We are still struggling to create actionable tools for the health sector to respond to known climate-sensitive diseases like mosquito-borne dengue and malaria. But there is much that can be learned from our past work to develop tools and partnerships to tackle these diseases. An event such as this can help us to build a community of practice, identify capacity gaps and needs, promote a culture of open data and open science, and develop climate services and tools for the health sector. And I'll mention briefly 3 examples. Many of us are working with the WHO and PAHO to develop an open online course called Climate-Informed Decision Support Tools for Public Health, an online course for climate and health practitioners and stakeholders. In addition, the IAI is working with the Belmont Forum and others to mobilize global funding through collaborative research actions on climate, environment, and health. And finally, in 2021, the IAI will be hosting several regional workshops in the Americas on climate, environment, and health to build transdisciplinary science policy teams to address these issues. So I am optimistic that this conversation will help us to move forward to tackle the challenges that we face as a global community. Thank you.
Thank you, Anna, for these very insightful comments, which I think they add an important dimension that should be taken into consideration. Obviously, other infectious diseases also still exist. They have not vanished, and so we need to take them into account also, among many other things that you suggested. So thank you very much. We now move, we'll have the questions after the four speakers. So we now move to Akimasa. Akimasa Sensei, it's your turn. Thank you.
Okay, thank you for your kind introduction. My name is Akimasa Hirata. Basically, I am a computational modeler for physical agent to human health. But for this COVID-19 crisis, my group is interested in evaluating the effect of meteorological factor on morbidity and epidemic duration of COVID-19. Having reviewed some related paper, there are so many papers related on this topic, but their findings were not consistent, and I believe this topic is still controversial. For example, Chinese group reported 122 cities. Their investigation found no clear relation between the ambient condition. At that time, main factor was temperature and humidity. Instead, time series analysis of Italian cities, they found some correlation with meteorological factor, temperature, humidity, and wind speed. But for each city, not in a multi-city analysis. So it is very difficult to characterize the transmission characteristic in different cities measured at a different way, I think. It is so that this makes it difficult for compare or to derive a straightforward conclusion. So my group is conducting the Japanese case studies considering the 90 prefectures. Feature of Japanese study is the uniformity of the policy, health insurance system. It is free, basically, and household wellness is almost uniform. But unfortunately or fortunately, the number of cases reported in Japan was limited, at least in the first wave. For example, in Tokyo, the population of Tokyo is 13 million, but the reported cases in the first wave was 5,190. So for relatively small number of cases, it is also the effect of nosocomial and nursing facility infection was not always reported. So this may be an uncertainty factor in very small numbers. In some of the prefectures, time evolution was a series of cluster infections. So several cluster infection was observed when the people from foreign country come back to the Japanese. For example, for the travelers in the maybe China and other countries. So I tried to simulate this kind of transmission by computer, but it didn't work so much. Unfortunately, due to such cluster infection was dominant in Japanese first wave. So all we can do was actually to conduct a statistical analysis as many people has conducted. So we cannot find the correlation between the ambient temperature condition and the morbidity rate first, but After considering the effect of population density, we found correlation with morbidity and mortality rate. So my tentative perspective is to consider the social distancing characterized by population density is important, but still meteorological factor may not be neglected. This is my perspective at the moment. Thank you for your kind attention.
Thank you very much, Akimasa. I think you introduced some very important topics that I expect that will stimulate discussion, such as mobility, social distancing, and also a bunch of statistical limitations having to do also with the underreporting of cases and all the errors associated to case data and so on. So I think that these are very interesting remarks that we should keep in mind also for prediction purposes in the discussions of tomorrow. So thank you very much. Now we move to Mercedes Pascual. Mercedes, the floor is yours.
Yes, hello everybody. It is a pleasure to be here. Now, given the limited state of the evidence so far on the role of climate factors on COVID-19 transmission, I will focus my short remarks on what could be done. So first, I think it will be productive to address two related but separate questions. One, whether climate factors influence the timing of emergence, and two, whether they influence the seasonality of the disease. The former question should be less confounded by other factors. Now, second, for the latter question, we do know from other emergent epidemics, like that of cholera in Haiti, that the effect of climate factors on seasonality are typically absent in the first outbreak, even though they can manifest themselves very clearly in the following seasons. In the example of cholera, the phase of the season relative to rainfall established in the second wave, not the first. And this is consistent with what has been said we should expect for COVID-19, where the large fraction of the population initially without immunity would preclude a clear effect of climate factors on the seasonality initially. Now, third, if seasonal drivers do operate, their interplay with levels of immunity will become important to determine the size of successive peaks and the times of reemergence of large outbreaks. We have recently shown in work on the transmission of another virus, namely dengue, that for infectious diseases with relatively low average reproductive number, R0, prediction of time to reemergence can be extremely sensitive to values of this number and to the fraction of non-immune individuals after the first wave. Now, COVID-19 may live in this space, which sets an intrinsic limit to prediction. To address their interaction with immunity levels, climate factors would have to be considered within transmission modes. So fourth, we should be able to infer the role of seasonal factors and incorporate those into epidemiological models based on now the temporal surveillance data collected across different locations. There remains, however, too much uncertainty still on the basic epidemiology inferred from case and death data with such models. Inference should be possible, but not without concomitant information on the levels of testing, as this influence the case data and also the death data. And in our own work, we have recently shown how central it is to consider the testing as part of the mapping from infections to reported cases in these dynamical epidemiological models. Without this, even the definition of asymptomatics becomes problematic. So in brief, we can be in a good place to do inference and especially to take advantage of parallel data across space at a regional level, as long as we do include information on testing that is essential to map the cases to infections, as well as data on serology. Finally, from the work on other respiratory viruses, in particular on influenza, we can expect that explanations of seasonality will differ in going from temperate to tropical regions and at different spatial scales. And preliminary results on this front can be seen actually in the ongoing work of Xavier Rodo and his group, in the poster by Forlan and colleagues, where statistical evidence for effects of absolute humidity and to lesser extent of temperature are presented in a global analysis.
Okay, thank you, Mercedes. Thank you for the mention too. I think you raise a bunch of extremely interesting ideas that we'll need to discuss more and more, because maybe not only today, even though the timing, I think you are doing quite well as a speaker, so you really constrain a lot to the timing. We'll have some time for discussion, but I think that these are issues that will be of much interest also for the prediction panel. So the role of low levels of infection and how these can generate spikes, new spikes, and whether what we are seeing in some places might be assimilated to that. I think you also made a bunch of other interesting topics, but let's move to John and let's leave all that for the questions and answers. So John, it's your turn. You are the closing speaker for this panel, so thank you.
Great. Thank you so much, Javier, and thank you to my colleagues at the NOAA, at AGU, and all of the organizers of this conference. I'm very honored to be included on this fabulous panel. In thinking about my comments, I was reflecting, this is truly an unprecedented pandemic we are in, almost unprecedented. I thought back 100 years to the 1918 H1N1 global influenza pandemic and try to think about how we would answer this question. We've seen the graphs of the differential responses to that pandemic in different cities of the United States. And I was just struck by the dizzying array of scientific capabilities that we now have to be able to ask this question about what are the influences on transmission, especially what are the environmental and climate. Just thinking through these things, 40 years after 1918, we were able to describe the structure of DNA, and 50 years later, we not only have the whole human genome, but we were able to sequence the SARS-CoV-2 virus by January 11th, and now we have real-time sequencing of the virus. We have satellites that are capable of capturing everything from temperature to humidity to lights at night to be able to describe human behavior. We have high-speed computers capable of running remarkable population simulations and other kinds of high-speed computer modeling. And we have electron microscopy and a whole host of other laboratory capabilities to allow in-depth study of not just the genome, but the structure, the function of the virus. So I think with all of these capabilities, we should be asking the question, how do we deploy all of them in an integrated fashion to be able to best answer these questions? So, I'm a doctor, so I'm not really trained as much, but I'm kind of trained as an epidemiologist and a toxicologist as an occupational physician. And the epidemiologist in me says, well, we have a global pandemic, we're doing global scale ecological studies, we have a massive study population, we should be able to detect some things, but as we've heard, there's a lot of problems with bias and confounding that limit that. As a toxicologist, I say we have all these exquisite lab capabilities. We can measure the survival of the virus in a lab in all kinds of different settings. We could even look at its conformation of its spike proteins if we wanted to in different settings. But we really have to have that real-time affirmation. So I want to make this case for integrated looks at all different lines of evidence, and it kind of reminds me of some of the work that we're doing at the National Toxicology Program about systematic review of the literature that is trying to figure out how do you combine the evidence, a systematic review of 30 epidemiologic studies with the toxicology studies, with the electron microscopy studies. So what do we have? What do we have with all of this scientific capability? We have these multiple ecological studies. As we've been discussing, there is this suggestion of an influence of temperature and humidity, but these studies are all very limited. I just want to say the study that Rachel was describing earlier looks like one of the best studies for controlling so many of those variables of interventions, public health interventions that are hard to model. We have laboratory studies. These are also showing some influence that there might be an optimal temperature and humidity range for survival on surfaces. we still need a lot of work to do, I think. I haven't seen the studies looking at ventilation systems. We're just getting into that. But that's obviously hugely important because there's a lot of evidence that some of the super spreading events are taking place indoors, where the influence of outdoor climate and environment are at least translated by a ventilation system. We need to understand that. And we have simulation models that are providing a lot of insight into the relative driving force of these environmental factors versus the other behavioral, the compliance with the public health interventions and the other interventions. I'm thinking of the Baker et al. Study, which I think has influenced a lot of the thinking on this. So I would propose that we think about how we integrate these lines of evidence. How do we identify the best quality epidemiology studies, and we can use those to start to bound some of those modeling parameters. We saw, Rachel's look to come up with an effective R0. I think that is the way to go, is to be able to inform scenarios of different R0s, and then we can look to multi-model comparison studies, just like we do for climate, and use the best available sophisticated models and integrate these lines of evidence, as well as I mentioned, a systematic review of the laboratory studies to try to give us the best available insight into the response of the virus to different kinds of environmental settings, whether that's outdoors in an air duct or on somebody's respiratory tract. So I'll stop there and look forward to the discussion.
Okay, thank you. Thank you very much, John. I think that the stressing of global capacities was an important topic, because one of the things that we need to come out from this meeting also is what to do next, and how we're going to mobilize these global capacities in the environmental and climate field domains. We have many agencies here. So how we can mobilize to help an effective response. So I think that that's among others a very important topic. But maybe before beginning the panel, maybe I would like to begin with a question for you, elaborating on what you mentioned, and that others can also chime in and comment. So imagine that we could conclude that SARS-CoV-2 and COVID-19 do have some meteorological or environmental sensitivity. What are some examples of how we might use that information in terms of disease response in implementation science that you can think of?
Well, if that question's coming back to me as the start, I'm sure the other panelists will have more informed ideas on that.
Anyone can
I think what we've been hearing is that right now the behavior of the virus and the interventions needed to control it are being driven by the number of susceptibles, and that with such a high number of susceptibles and such a transmissible virus, that the climate and meteorological influences are relatively limited, and you can't really base a control strategy just on those. I think that we may be able to give some advice over time on the indoor versus outdoor as we cycle through the seasons and we start to understand the relative importance of those settings for transmission. We may be able to give some advice, but I think that it seems like we're in a phase now where it's really about behavior, it's about personal protective equipment, and ultimately it will be about other kinds of interventions like vaccines or other prophylactic medication treatments, and that the real role, as we're hearing of, I think Mercedes was saying this, of the seasonality and the real driving force of the meteorological factors will be seen, once the population susceptibility goes to a much lower level and the R0 goes down closer to 1.
Anyone else wants to say something about this issue of implementation science? So can we learn from examples from other diseases where this climate information has been successfully integrated that we might think of for COVID-19?
Hi, Javier, I can comment. If we were able to make that assumption that there is a climate signal, then I think that would be a place to move quickly with our health sector partners to begin to develop climate services, different tools, outputs, depending on their needs, whether in the format of maps, bulletins, different ways of transmitting this information, and ideally through forecast models, early warning systems, so that they have sufficient lead time to better allocate resources and personnel, to purchase laboratory diagnostics, to issue education and alerts to the communities. That's sort of the general approach for developing climate services for infectious diseases that has been explored with other types of climate sensitive diseases that could certainly be relevant in this context, perhaps would inform the timing of vaccine campaigns, for example.
Yes,
Okay.
I agree. The question of,
okay, Mercedes.
Sorry, go ahead, Javier.
No, just say what you have to say.
No, I just wanted to say that although we may see the effect of climate more clearly in a second wave, we can start looking for the signal of that, as I said, in the perhaps less confounded timing of the initial emergence or that we saw across the globe. But for the seasonality, the question of how much build up of protection in the population we need to see to start being able to detect the role of environmental factors on seasonality is an open question, and I think it's a very important one to keep to basically be prepared to ask as we move forward. It may not. And I didn't want to mean that we need to basically be at very, very low levels of non-immune individuals for this to become clear. I think the models can tell us.
Okay. So we'll move to some of the pre-specified questions that the committee elaborated for our panel members, and then we will pick the remaining questions from the Q&A in the chat. So I'd like to organize these in different blocks. So I think that one interesting, because I think this is interesting to tease out for other sessions, the days after. One is what do we learn from the theory of other diseases? So that might be one thing. Then also about first waves that you also commented on, some of you. The role of seasonality and why is it important, and also the environmental modulation and why that can be a factor that might be relevant. And I wouldn't like to end the session without having a discussion on the uncertainties of data quality and the methodologies and what these might preclude at this initial stage of the pandemics. No? So let's begin by the theory, and one of the questions that I think it's quite trivial that I had to pose to you is, at the moment, even though you had somehow answered this in a way, is how much can we infer about COVID-19 sensitivities from our current knowledge about environmental drivers for the respiratory viruses and diseases? And are there specific facts about these other diseases that you think are likely to apply also to COVID-19? And where do these analogies break down? So whoever of you wants to answer. Maybe it's a long question, so you can concentrate on the analogies and disparities. So how do you feel about that in comparison to other diseases?
Well, if there's silence, I will jump into it. I was doing a literature search on that as I was preparing my comments, trying to see how much intercomparison there's been at the laboratory level. And I haven't seen much of that, although I'm sure it exists somewhere. What I did find was an intercomparison of the epidemiological characteristics. So we've all seen those, the comparison of SARS-2 to SARS-1 and to MERS, and the fact that MERS had a very, very high case fatality ratio, a very low R0, and kind of did not have a global pandemic spread. The same thing to a lesser extent, but same kind of outcome with SARS. So those kinds of comparisons are interesting, but as we're saying, we don't really have a full handle on the why SARS-2 is behaving differently from SARS-1 and MERS in terms of its much higher R0 and global spread in a very short time. I think we're seeing some really intriguing molecular evaluations. The conformational change in the spike protein that seems to help it elude immune systems, is an interesting piece of that. We have to start looking whether those conformational changes then are influenced by humidity, by temperature, et cetera, to get real deep into the mechanistic side. And then as we're doing, we're also doing this kind of macro modeling approach to understanding these differences too. And again, I would just make the case to integrate these different kinds of lines of evidence.
So if there is no other comment, we move to the second question. In your estimation, has SARS-CoV-2 and COVID-19 dynamics exhibited any evidence of seasonality or other sensitivity to environment, meteorology, or climate that you believe in at the moment? Just take into consideration that there is press also in the conference, so they want, maybe it's trivial and it's something that they want to hear about. Or if it is not the case, how much or how long it would take to have an idea whether this seasonality is really relevant for COVID-19, even though we have not completed one full seasonal cycle, obviously. Who wants to answer? Akimasa?
Yeah, the seasonality, but due to the, for example, in Japanese case, the COVID first wave was from March to May. The data is actually very limited to conclude something. But from that kind of limited data, what we can found in our research was the percentage of population density contribution was 70 to 80%. So this is approximately the social distancing, but the remaining was characterized by the environmental factors. So the temperature, humidity, but they are, as you know, they are closely related to each other in very limited time period. So we cannot conclude at the moment, but I believe, all I can say is I believe there are some factor affecting the COVID-19 spread or transmission.
Mm-hmm.
Yeah, yes, I agree with what was just said. I think there is some evidence, and we see some in the posters that are here and in some of the preprints that have been mentioned, but there are also some contradictions. And I think the very important control of more uniform conditions of interventions, behavior, sampling, will have to be taken into consideration. This is why also, given the limitation on the length of time, we have to take full advantage of the multiple locations. But we can only do this if we can control for other things that vary across these multiple locations. And this is where this ancillary information, in particular on the measures that affect human behavior, are going to be very important, but also on the sampling. And I think I'm sort of stressing this because if we don't consider that, we don't even have a dependent variable. And the question of whether we can look at RT as the dependent variable, we have to be a bit careful, because RT is effective exactly because it takes into consideration variations in levels of immunity. So as we go on, making it a dependent variable could be problematic.
So in this regard, how Mercedes and others, how can we think of a proper analysis that takes into consideration all these limitations and also the scale at which the analysis is done? Because on the one hand, it's like having the fantastic result is that showing globally. But then we know that aggregating data at these initial stages where there are so many uncertainties might be very problematic. So we need to resort to local behavior and local dynamics, how to make this interplay reasonable in an attempt to uncover these relationships. You are muted, Mercedes.
Yeah, sorry. I think researchers are already doing this, but certainly you point to a very important issue of focusing on regions and on variation within regions and how we define the proper spatial scales, depending also on aspects of the regional climate, aspects of the different interventions and the different situations of the different countries. I mean, we know this even for influenza. We understand very well seasonality in what concerns temperate regions. I think for tropical regions, it is much more complex. So I think there is a hope to do this globally, but with considerations within regions. And I think Rachel pointed out some approach, for example, some statistical approaches that could do that.
Mm-hmm. Okay, so linking with that, Akimasa, how you think that errors should be taken into consideration at these initial stages, because it might compromise results. So in which sense we should try to integrate that uncertainty in any diagnostic, but also prediction attempt.
So to suppress the uncertainty, actually, I have tried to take into account the window effect, for example, but the measurement of window is not stable at all, as you know. So this kind of uncertainty is not well considered in my research. And in Italian group conducted some research for to evaluate the effect of wind. At that time, in time series analysis in one series, very consistent result was obtained. So how to collect such kind of finding and then accumulate to suppress uncertainty? It is very challenging, and actually I have no exact idea to how to cooperate G1. And my group is doing the multi-country analysis at the moment. I couldn't get very consistent finding about that.
Mm-hmm.
Yeah.
Okay, we have a question here by the committee on stemming on Dr. Bernstein keynote speech earlier today, and on this interaction between COVID-19 and the environment, and how humans have altered the environment. So the question posed is whether you see associations in general terms between epidemics and global change. So either specifically for COVID or in your work on other infectious diseases. And that might be relevant also in the setting that we are now.
Well, maybe if it's a very general question, and I think the very good first talk today addressed this. Of course, it's a very, there is, I think, convincing evidence of the increasing frequency of emergence, of spillover, etc. and the relationship to environmental degradation. So essentially creating the conditions that facilitate this kind of event. So I think that there is sufficient evidence on that front to act. We can and we can give more examples than what was mentioned this morning, but I think we all would agree on that. I mean, the roles of deforestation, land use change, the way we produce food, I think we have to consider all those factors, in particular in the context of climate change and this increased connectivity of the world. I think it's madness not to do so.
Just to add on to that, I think that the evidence that has been provided so far has been stronger for diseases like the Zika viruses and Ebola viruses and those that maybe don't have quite the same intensive respiratory transmission. I think this is an area, spillover, understanding the interaction between land use change, poaching, human behaviors, and climatological factors and El Niños, et cetera, is relatively understudied. I think we could be doing a much better job of understanding this and being proactive in our stance and setting up the early warning systems that might, lead us to a One Health informed, proactive, monitoring stance where we are more actively looking for spillover events or the potential for spillover events in hotspots that get created. This is an area that we're talking about within the U.S. Global Change Research Program. We're trying to see if there are ways to build this capacity. And I know that USAID has just started a follow-on to one of their projects. It's called Project Spillover, to try to invest a little bit more in understanding this phenomenon.
Okay.
And I would add to that also the need to consider multiple epidemics that are now co-occurring that have interrelated yet distinct environmental, climate, and social drivers. For example, what's happening in the Americas right now with dengue fever and COVID. For example, this year, Argentina, which is at the southern edge of dengue emergence in the Americas, experienced the largest dengue epidemic ever. And yet that was, I think, been largely overlooked. And so how are these interacting? How are they affecting the same or different vulnerable populations? And again, this should be understood in the context of unequal social conditions. So as these global change processes accelerate, we tend to see the most vulnerable populations who are facing the greatest impacts and the greatest burden of disease. And these epidemics can reveal but also exacerbate these inequalities.
Okay, so we can move to the next question. Just I think that for the audience, it would be good to separate among the effects on the pathogen and the effects on the propagation of the disease, because it's not exactly the same and it's very different, you know? So you can have the pathogen but not have the disease, and so on and so forth. And the effects of environmental modulators can have a clearly distinct impact on those. So I think it's good to make this statement. Now we might move to the questions. So for this, I have the highly esteemed help of Mark Shimamoto. So he will be making the triage for the panel. So please,
Javier, could I just make one quick comment, just in line with what you just said about distinguishing between effect on transmission and effect on disease severity. We've been focusing a lot on seasonality and climate and the meteorological aspects, but I do want to say that the line of evidence for the impact of air pollution on modifying the severity is in a different place. I think it's probably a little stronger, in part because we have this very, very long background of evidence about interaction between air pollution and infectious diseases. Again, I think there's an integration of lines of evidence. There's a lot of molecular level evidence with the angiotensin converting enzyme receptor and its modulation by both underlying disease states like asthma and treatment for asthma, and the difference between the different air pollutants. So and that really gets to me, that modulation of severity gets to the point that Anna made in her opening comments about the social aspects and the social injustice aspects. And I think there is information that can be acted on now with regards to that. As opposed to what we're saying about the seasonality, where perhaps that's going to play out when we shift into an endemic phase. I just wanted to make that distinction.
Thank you for stressing.
Well, I'm going to state that it's incredibly important for everyone to know the difference between the virus and any potential disease. It's a fundamental principle of infectious disease that viruses and diseases are different things. And we have to look at how climate impacts each of those things separately and together.
Thank you, Eileen. Yeah, fundamental point. So, Mark, did you have a chance to choose some of the relevant questions? I think there are many of them.
Yeah, there were a lot of great questions as well as active discussion in the chat. Some of you were responding to others' questions, which is always great to hear. The first one kind of revolves around this issue of behavior, but one of the questions was around culture and habits and how that kind of influences COVID transmissions. Much we know, and we didn't quite take a deeper dive, but we thought maybe with your past experience in different outbreaks, what you've seen in terms of how behavior and cultural behavior influences transmission that might be learned when we're looking at the environmental determinants for COVID-19.
Maybe you can point to someone specifically.
Yeah, maybe we could start with Anna, if she had any thoughts on cultural and social dimensions of that.
I think it's fundamental, right? And when we're talking about diseases that are infectious diseases transmitted from person to person, how people interact, the proximity, whether we're in Latin America and hugging and kissing is the norm or not, will have a huge impact on transmission. So I don't think it can be overstated. And also it's hard to generalize. And that's why there were some comments about how to make recommendations for different social interventions. And my comment was, well, it depends where you are. If you're in, a country like, in Brazil, you have urban communities, you have Amazonian indigenous communities. Those interventions need to be tailored for the different communities and their own social and cultural norms. So it's difficult to make a blanket wide recommendation.
Any other thoughts?
Let's move to the next question, Mark, please.
All right. So the next question was kind of digging a little deeper, and John kind of highlighted this in terms of air quality a few minutes ago, but maybe pulling apart the nuances between airborne transmission from meteorological factors with more droplet level transmission, which could often be more person to person, and how that could play out both in terms of differences, but also how that could be influenced in indoor environments versus outdoor. So it's a little bit of a broader question, but if anyone wanted to speak to an element of that.
I mean, I'll just say that I think that question just opens up a Pandora's box of questions and uncertainties and things that we don't know. I mean, I think we saw play out in real time this uncertainty about the role of droplets versus airborne spread and people making assumptions based on that. I think it seems pretty clear now that both are modes of spread and that in fact the airborne non-droplet, very small particles may be very important in indoor settings where we're seeing these super spreader events. But what I haven't seen yet, and maybe it exists, is then asking these secondary questions. What's the role of a clean filter versus a dirty filter? What filter is best in an HVAC system? What about other biologicals in an HVAC system? Are there interactions? There are so many questions that come into play, let alone, indoor air pollutants, nitrogen oxides, ozone versus outdoor air pollutants. I think it just opens up, the more ways there are to spread, obviously, the more questions that come, and the more importance we play on the indoor environment, the more it just points to our relative ignorance about indoor air quality and indoor environments.
So linking with that, I think that's a very interesting point, John, because I'm also working with a group of biomolecular biologists trying to extract the virus SARS-CoV-2 from aerosols. And so far, this is an international initiative, and it's been extremely hard and extremely difficult. So now it seems to be consensus that there is an airborne transmission. And on the other hand, it's extremely complicated to get a number of copies where you can say that it is not just mere contamination of your sample. So is that because we are seeing from the epidemiology of the disease that there must be something that justifies this airborne transmission? And what is it?
I think that's a question I can't answer with my depth of understanding and exposure to the science. It seems like it might be the case, but I really don't know.
Okay, so maybe that's a question also for the modelers tomorrow. Does that justify adding a compartment and an extra compartment just to model this extrinsic behavior, also linked to the airborne potential transmission? So I leave that for the discussion. Okay, Mark, we have time for a few more questions.
Yeah, we did just get a follow-up question from Neville that says, again, similar to the discussion, but looking more at the relative of droplet transmission versus what does airborne mean relative to droplet transformation? I know that's a little bit more specific, but I didn't know if anyone wanted to tease that out more.
Question there is a little unclear. What is the question?
Read the question.
Yeah, Neville.
Yeah. Thank you.
So
the question really is, I understand airborne. I want to understand the distinction between what it means to be an airborne transmission process versus a non-airborne droplet mediated process. As I understand, it's really to do with the size of the droplet. So where's the distinction between airborne and droplet transmissible diseases?
yeah, I don't know the exact definition of airborne, but I think in this community, we assume the airborne transmission is actually indoor and confined space. It remains in flight for 1 hour or so, I think. But actually, I don't know the exact definition. Yeah.
I mean, Neville, I think again, I'm not the expert on this, but my understanding is that there isn't a clear distinction, but there are some size cutoffs for what we call a respiratory droplet versus a smaller size particle. There is probably a continuum that's generated, that the smaller the particle is, as Akimasa is saying, the longer it is likely to stay suspended in the air. Droplets, because of their size, tend to settle out more quickly, and so that has implications for not just the size of the filtration in a mask or a respirator, but also for distancing, also for surface cleaning, also for how do you behave in terms of interactions and social distancing, both indoors and outdoors. And I think that Javier, your challenge with your efforts to extract the virus from smaller size particle reminds me of work that I was involved in 10 years ago, you know, when all the complications of trying to understand nanoparticles. And I think, you know, maybe this is just one of those areas where we're at a scientific frontier, and we are developing the techniques at that small scale for understanding aerosols.
Yeah, I think it's a completely complementary panel discussion that probably is not the expertise of or not the main expertise of the members here. So maybe it's extremely important and relevant, obviously, and as you say, let's think on that as a way of transmitting the virus, not the disease. That is a different. I'd like to make those points again because it's something important to make, and also the fact that for different communities this might mean different things. But let's leave that, and maybe let's go back to the role of climate. And so obviously pollution and the way by which the virus might be circulating in droplets and aerosols has to do with temperature and humidity conditions. So this is something that go hand in hand and will have to be also tackled. But maybe we can leave that part of pollution and just concentrate more on climate questions. Just if I can say climate is something separate, which I'm not saying now. Okay, Mark, go ahead, please.
I think for time's sake, we're going to pass it back to you to close out this session, and then we can close out the day.
Okay, so thank you very much. I think this was a very fruitful discussion. Obviously, it reflects the state of the art of the problem. It's a new disease. It's something that it's coming. We are reacting as fast as we can as a community, and I think that there are many interesting insights that you've made, and many lessons from both COVID-19 disease dynamics, but also from your experience on other infectious diseases and how we can gain understanding and anticipation capacity. So I pass now the floor to Ben to just wrap up this first day, and I thank you, the four of you, for your participation in this interesting panel. So Ben, please.
Great, thank you so much, Xavier. I'm at my own Zoom moment here and cannot get my video back on, but that doesn't matter. So my thanks to really everyone for attending and participating today. We're closing now. This is a global conference, and for those who are doing the math, Akimasa just tuned in for his section at midnight his time, and so we need to end now, and also we have colleagues even further afield who are in the middle of the night for this. But it's really a 24-hour event, right? So the iPosters are available. Some people are available for the hours of the conference. If they're not available, you can always message them so we can keep this conversation going. The AGU Connect site, and I appreciate that there's a lot of technology we're juggling here, but in the original email people received, there is a link to that online discussion forum, the AGU Connect site, where we already are having some active discussion, and we can continue to have conversation there. We will attempt to get unanswered questions from this session up onto that site as well. But with that, we will close the live session, and then tomorrow, beginning at the same time, so I'll let you do the time zone math for yourself. You're seeing on the screen the time that we will be beginning in the US Eastern Standard Time, the 1 p.m. Greenwich Mean Time. We'll begin a discussion of really trying to build from what we learned today about understanding into the problem of forecasting, risk monitoring and forecasting. Where are we? Where do we need to go with that from the perspective of meteorological, climatological, and environmental drivers of this disease? So again, thank you so much for your participation, and I look forward to continuing the discussion overnight and then tomorrow. Take care.